So we're GeneQuine Biotherapeutics, and we're developing a novel gene therapy platform based on helper-dependent adenoviral vectors. As you may know, gene therapy is quite a hot field currently because it potentially offers a cure or transformative treatment for chronic diseases. And some of the most commonly used gene therapy vectors include AAVs, mostly for in vivo gene therapy, and retroviruses, lentiviruses for ex vivo gene therapy. And there are actually a number of companies who have successfully built robust platforms based on these vectors. Examples include uniQure, Spark, Oxford Biomedica, and so on. And we want to do the same now for helper-dependent adenoviral vectors. And helper-dependent ad are just as good in terms of long-term gene expression as the other vector types are, but they offer the additional benefit of having a large DNA packaging capacity of up to 36 kilobase pairs, which means that you can insert a lot more DNA into these vectors. For example, AAVs have only a packaging capacity of 5 kilobase pairs. So that gives us a lot of freedom in the design of the vectors. We can insert large genes and also multiple genes into one single vector. And also, helper-dependent adenoviral vectors do not integrate into the host cell genome, so that means there's no risk for insertional mutagenesis, which can be problematic with the other types of vectors. So that's why we believe that helper-dependent adenoviral vectors are at least as good as the other types of vectors, and for certain indications, they're superior. So in our first development program, we're developing disease-modifying agents for treatment of osteoarthritis. And osteoarthritis is the most common joint disorder with a huge unmet medical need, because currently there are only symptomatic treatments available, but no disease-modifying treatments. So no treatments that could affect the progressive cartilage degeneration, which forms the basis of the disease. So one of the major challenges in treating joint diseases such as osteoarthritis is the question, how do you get your drug into the joint efficiently? And if you administer the drug systemically, the drug needs to exit the capillaries in the synovial membrane and then diffuse through the extracellular matrix in order to get to the joint fluid, which is a very inefficient process. It's possible to some extent for small molecules, but it's very difficult for large molecules. And then you can obviously just inject your drug directly into the joint, intra-articular injection, but the problem with that is that the lymphatic system of the joint will clear out the drug quite rapidly. That's a size-independent process, so it's true for small as well as large molecules. And that is obviously a major problem if you want to treat chronic joint diseases such as osteoarthritis. And we believe that the solution to the problem is to establish long-term protein production inside the joint. And we do that with a local gene therapy approach using a helper-dependent adenoviral vector expressing interleukin-1 receptor antagonist. And the way it works is that the drug is injected directly into the joint. It distributes in the joint fluid. It enters the synoviocytes, so the cells that surround the joint capsule, and those cells produce then interleukin-1 receptor antagonist, which is a secreted protein, so it gets into the synovial fluid again, and then it blocks the interleukin-1 receptor, and thereby we stop the vicious circle of inflammation and cartilage degeneration, which is ongoing in osteoarthritis, and at the same time we also inhibit pain. So with a single injection, we can establish long-term protein production, leading to symptomatic as well as disease-modifying therapy. We've shown that in mouse joints, when we inject them directly into the joint, we can establish long-term production for more than 1 year with helper-dependent adenoviral vectors. We've also shown that we can have a disease-modifying effect in a mouse osteoarthritis model, shown here by the significantly higher cartilage volume in the treated joints compared to the controls. And then we've shown in a horse osteoarthritis model that we can treat pain, the major symptom of osteoarthritis, efficiently. On the left, you see a horse that was with osteoarthritis was treated with placebo, and you see the leg cannot be bent completely because the horse has pain in the joint. And on the right, you see a horse treated with a vector, and you see the leg can be bent completely. That's because the horse is pain-free in the joint, just as a normal horse would be. In the same model, we were also able to show again that we have a disease-modifying effect, shown here by the significantly lower scores of the cartilage histology scoring compared to placebo. And in fact, the values were kind of comparable to those of normal, healthy joints. So in summary, gene therapy can overcome the delivery limitations that you have with conventional therapeutics in osteoarthritis. And we've been able to validate helper-dependent adenovirus vectors for joint gene therapy. We've significantly de-risked the development of our lead product, GQ203, which is a helper-dependent adenovirus vector expressing interleukin-1 receptor agonist, by demonstrating symptomatic as well as disease-modifying efficacy in small and large animal models. And now we have also the potential to develop further product candidates based on helper-dependent ad, taking advantage of the unique features of this vector type.